Okay, let's unpack this. We are diving into enzymes, really the engine room of biochemistry. And for you, as you're preparing for your MBBS journey, getting a grip on these molecular machines isn't just, chemistry. It's absolutely foundational to understanding pretty much every physiological process, every medical diagnosis you're going to encounter.
That's right. They're central to everything.
Our sources today, well, they give us a really comprehensive, conceptually strong overview. We're going from the basic definition of a biocatalyst all the way through kinetics, regulation, and crucially, their role in clinical diagnosis. Our mission here in this deep dive is basically to shortcut your understanding. We want to connect the molecular nuts and bolts directly to what you'll see at the bedside.
Making that connection is key.
So let's start right at the beginning.
Well,
What exactly is an enzyme? Our sources define them pretty clearly as biocatalysts synthesized by living cells. They are overwhelmingly protein in nature. We always have to mention the exception, of course.
right, the ribozymes. RNA molecules that can catalyze reactions, usually on themselves. But yeah, the vast majority are proteins.
And beyond that protein core, they've got some key properties. They're colloidal, they're thermolable, meaning, sensitive to heat, and they have this incredible specificity.
That word catalyst is crucial, but sometimes it feels a bit understated, doesn't it? You have to appreciate the sheer power here.
How so?
Well, think about the difference they make. In a
Yeah.
chemistry lab, if you want to do something relatively simple like protein hydrolysis, breaking down protein with strong acid at 100 degrees Celsius, it takes days, literally days.
Right, harsh conditions, slow process.
Now, contrast that with your own body. Full protein digestion happens at just 37 degrees Celsius, normal body temp, and it's done in a few hours. That remarkable difference, the speed, the mild conditions, that's purely down to the enzymes in your digestive system.
It really is astonishing how they manage these reactions at such low body-friendly temperatures. The sources use a great analogy, actually. The enzyme is like a teacher,
Ah, yes, I like that one.
making a really difficult textbook subject, that's the substrate, understandable and, processed quickly. It just ramps up the reaction rate, the learning speed dramatically.
Exactly. And just to put a bit of history on it, while Berzelius came up with catalysis in general terms, it was Wilhelm Kuhn back in 1878 who specifically coined the term enzyme. It literally means in yeast because that's where some of the first observations were made.
In yeast. Makes sense.
But the real breakthrough, scientifically, confirming they were actually proteins,
Mhm.
that took a while longer. It was James Sumner in 1926. He managed to crystallize the enzyme urease from jack beans.
Crystallized it.
Wow. And that crystallization proved its protein nature, which was actually quite a debated topic back then. So if we connect this back to the bigger picture, the real reason enzymes exist, their core function,
Yeah.
it's incredibly powerful. They make life at normal body temperatures possible.
By tackling that energy barrier.
Precisely. They dramatically lower the activation energy. That huge energy hump reactants normally need to overcome to become products. Enzymes just slash it.
They find a shortcut.
They essentially build a tunnel through the mountain, reducing the barrier height without changing the starting or ending energy levels. That's absolutely key.
So they speed things up, but they don't change the overall energy difference between start and finish.
Exactly right. They don't alter the thermodynamics, the equilibrium. They just affect the rate at which equilibrium is reached. They build that tunnel, making the journey much, much faster and easier. And that's why all those complex metabolic pathways in your body can hum along efficiently below 40 degrees Celsius. Without enzymes, nothing would happen fast enough.
Okay, so we've got the enzyme as this essential protein machine, lowering activation energy. Let's transition now into section 2. Enzyme structure and classification. I mean, there must be thousands of enzymes in the body. How on earth do we organize them all?
Right, it'd be chaos without a system. We rely on a really systematic nomenclature established by the International Union of Biochemistry, or IUB. And the clever part is, it categorizes them based on the type of chemical reaction they catalyze, what they actually do.
Ah, function-based. And that leads to the EC number, right? That 4-digit code.
Exactly. The enzyme commission number, 4 digits. The first tells you the major class, the second, the subclass, third, sub-subclass. And the fourth number identifies the specific individual enzyme.
So if you know the EC number, you know exactly what reaction that enzyme is responsible for.
Pretty much. And for your MBBS foundation, getting a handle on those 6 major classes is absolutely paramount. The mnemonic people often use is OTHELIL.
Oh, ALIO.
Yeah.
Let's quickly walk through what each class does. It gives you a great map of biochemistry. 1, oxidoreductases. O. The electron movers. They handle all the oxidation, reduction, or redox reactions. Transfer of electrons, hydrogen atoms, think energy production.
Like in the electron transport chain. Precisely. Alcohol dehydrogenase is another common example. Cytochrome oxidases. Absolutely central.
Okay. O is for oxidor adductases. What's T?
To transferases. T. These guys are the molecular movers. They catalyze the transfer of functional groups, could be an amino group, a phosphate, a methyl group from one molecule, the donor, to another, the acceptor.
Like hexokinase in glycolysis, adding that phosphate to glucose.
Perfect example. Hexokinase transfers a phosphate group from ATP onto glucose. Very important, clinically relevant transferase.
Got it. OTH?
3-cragolase. Hydrolysis. These are the demolition crew, you could say. They catalyze hydrolysis reactions. That means they break chemical bonds by adding a molecule of water across the bond.
Ah, like digestion.
Exactly. All your major digestive enzymes. Lipase breaking down fats, pepsin breaking down proteins, amylase breaking down starch. They're all hydrolyses.
Yeah.
Use water to chop things up.
Makes sense. OTHL, first L.
4, lyases. Now, these are a bit different. They also break chemical bonds, but they do it without using water, unlike hydrolases. And often, when they remove a group, they form a double bond, or conversely, they add a group across an existing double bond.
So no water involved in the cleavage.
Right. Think aldolase and glycolysis. It splits that 6-carbon sugar, intermediate, into 2 3-carbon pieces, creating a double bond in the process. That's a classic lyase reaction. It's cleavage, but not hydrolytic.
Okay, subtle difference, but important. OTHLI.
Okansi, these are the internal reorganizers. They simply catalyze the rearrangement of atoms within a single molecule. They convert one isomer into another, same chemical formula, just a different structure.
So just shuffling things around internally.
Exactly. Like trisulphosphate isomerase, also in glycolysis, interconverting those 2 3-carbon sugars. It's an isomerization.
All right, and the final, LOTHLIL.
The legacies, L. These are the builders, the joiners. They catalyze the formation of new chemical bonds, linking 2 molecules together. Think ligate to bind.
And building things usually takes energy.
Almost always. Ligase reactions typically require energy input, usually coupled to the hydrolysis of ATP or a similar high-energy phosphate compound. Glutamine synthetase, which makes glutamine, as a key example, it joins glutamate and ammonia using ATP.
OVLIL. Okay, that framework helps a lot. Oxidor reductases, transferases, hydrolases, lyases, isomerases, ligases. Now we know their jobs. Let's look at the actual structure that does the job. The complete active unit. You called it the holoenzyme.
Yes, the holoenzyme is the whole functional entity. It's composed of 2 key parts, generally speaking. There's the apoenzyme.
That's the protein part.
That's the protein part, yes. It provides the main structure, the framework, and critically, it determines the enzyme specificity. It dictates which substrate molecule can bind.
And the other part?
That's the coenzyme, or sometimes a cofactor. This is the non-protein component that's required for the catalytic activity itself.
Okay, coenzyme, cofactor. What's the difference there?
Good question. Generally, cofactor is the broader term. It could be a simple metal ion like magnesium or zinc, we call those activators, or it can be a more complex organic molecule, which is what we usually call a coenzyme.
Right, and sometimes that coenzyme is stuck on really tightly.
Exactly. If that non-protein organic part is bound very tightly, maybe even covalently, to the apoenzyme, we call it a prosthetic group. In hemoglobin or cytochromes is a classic example.
And if it binds loosely?
If it binds transiently, just associating during the reaction cycle, we typically call it a coenzyme. Think of NAD+ or FAD. They bind, do their job, dissociate, get regenerated, and come back.
That distinction seems important functionally.
It is. Coenzymes are often described as second substrates or co-substrates, because unlike the main substrate, the coenzyme is chemically altered during the reaction. It might accept or donate electrons or atoms or groups.
So it participates directly.
Directly. It acts like a shuttle bus, transferring chemical groups, hydrogen atoms, electrons, amino groups, or acyl groups, you name it. And then it has to be regenerated back to its original form, often by another enzyme, to participate again.
Okay, and this is where it gets really interesting clinically, right? The link to vitamins.
Absolute critical link.
Here.
Many, if not most, of these essential coenzymes are derived from the water-soluble B-complex vitamins that you get from your diet.
Can you give some examples?
Sure. NAD+ and NADP plus EVA-T, key players in redox reactions, are derived from niacin, which is vitamin B3.
Niacin, okay.
Flavin adenine dinucleotide, FAD, and flavin mononucleotide, FMN. Also, redox coenzymes come from riboflavin, vitamin B2.
Riboflavin.
Coenzyme A, essential for acyl group transfer, like in fatty acid metabolism in the TCA cycle, comes from pantothenic acid, vitamin B5.
B5.
And thiamine pyrophosphate, TPP, crucial for certain decarboxylation reactions, Like pyruvate dehydrogenase comes from thiamine vitamin B1.
B1 thiamine. Wow, the B vitamins are basically coenzyme precursors.
They really are. Now, pause for a moment and think about this. Think about the clinical implications for you as a future doctor. What happens if the apoenzyme, that big specific protein structure, is perfectly fine, perfectly synthesized, but the coenzyme, that vital B vitamin derivative, is missing because of a dietary deficiency?
The whole enzyme doesn't work.
The entire holoenzyme is non-functional. The whole metabolic pathway that depends on that specific enzyme step grinds to a halt.
Ah, okay. That directly explains deficiency diseases.
Exactly. Think about pellagra caused by niacin B3 deficiency. Niacin is needed for NAD+. NAD+ is essential for hundreds of oxidoreductases, especially in energy pathways like the TCA cycle. If you can't make NAD+ NOD, those enzymes stop working, you block energy production, and you see the devastating systemic effects, dermatitis, diarrhea, dementia, particularly in tissues with high energy demands like the skin, the gut, the brain. The vitamin deficiency blocks the coenzyme, which blocks the enzyme function, leading directly to the disease state.
That connection is crystal clear. Structure, function, and clinical outcome all link through the holoenzyme and its parts. Okay, that detailed structural breakdown naturally leads us into section 3, mechanism of action and specificity. We know the parts. Now, how does this machine actually work? Where does the magic happen?
The action all takes place at a very specific location on the enzyme called the active site. The enzyme molecule itself might be enormous, a huge protein.
Right.
But the actual catalysis is confined to this relatively small region, often described as a cleft or a pocket on the enzyme surface.
And structurally, this active site isn't just a random patch.
Not at all. It's a masterpiece of molecular engineering. It's formed by a precise three-dimensional arrangement of specific amino acid residues, the catalytic residues.
Things like serine, histidine.
Exactly. Serine, histidine, aspartate, glutamate, lysine. These amino acids have side chains with chemical properties perfect for catalysis, donating or accepting protons, forming temporary covalent bonds, etc. And here's a key point. These catalytic residues might actually be very far apart from each other in the linear sequence of the protein, the primary structure.
But they come together in the 3D shape.
Precisely. The complex folding of the protein, its tertiary structure, brings these crucial residues together in just the right orientation within the active site pocket to interact with the substrate and facilitate the reaction. The source mentions lysozyme, where residues from positions 35, 52, 62, 63, and 101 all contribute to the active site.
That precise 3D structure must be why enzymes are so specific.
Absolutely. That unique geometry of the active site dictates the enzyme's specificity. And this specificity isn't just one thing. It operates on multiple levels. It ensures the cell's resources aren't wasted on unwanted side reactions.
Okay, what are the levels? The source mentions a few types.
Right. First, there's stereospecificity, or optical specificity. Enzymes can usually distinguish between stereoisomers, like L and D isomers of amino acids or sugars.
So L-amino acid oxidase only works on L-amino acids, not D-amino acids.
Exactly. Our sources mention the three-point attachment model as a way to visualize this. The idea is the substrate has to bind to at least 3 specific points within the active site in the correct orientation for the reaction to occur. Only one stereoisomer will fit perfectly like that.
Makes sense. 3 points define spatial orientation. What's next?
Second is reaction specificity. A single substrate molecule might potentially undergo several different types of chemical reactions, but for each possible reaction, there's usually a different specific enzyme.
So an amino acid could be, say, decarboxylated or transaminated.
Right, or deaminated. But the enzyme that catalyzes the decarboxylation won't catalyze the transamination, and vice versa. Each enzyme is specific for one type of reaction, even with the same starting molecule.
One enzyme, one job. Got it. And the last type, substrate specificity.
Yes, substrate specificity. This refers to how picky the enzyme is about the molecule it binds to, and this exists on a spectrum.
A spectrum. How so?
Well, it can be absolute. Some enzymes will only catalyze the reaction for one single specific substrate. The classic example is urease, which only acts on urea, nothing else.
Okay, very strict.
Then it can be relative or group specific. These enzymes act on a group of molecules that share a common structural feature or functional group. Trypsin, for instance, is a protease that cleaves peptide bonds, but specifically after arginine or lysine residues.
So it recognizes a feature, not the whole molecule.
Right. And finally, some enzymes exhibit broad specificity. They can act on a range of structurally similar substrates. Hexokinase, which we mentioned, can phosphorylate not just glucose, but also fructose and mannose, which are structurally related hexosugars.
It's absolute. Relative broad. Okay, that covers the what and where. Now, how does the catalysis actually happen? Let's talk about the theories. The old one first.
Right. The historical starting point is the lock and key model, proposed by Emil Fischer way back in the 1890s.
Lock and key implies rigidity.
Exactly. It pictured the active site as a rigid, preformed template, perfectly complementary in shape to the substrate, like a specific key fitting into its matching lock.
Simple idea, but maybe too simple.
That's the main critique. While it explains specificity beautifully, it struggles to account for the flexibility and dynamism we now know enzymes possess. How can a rigid lock explain, for instance, allosteric regulation, where binding at a different site changes the active site's shape? It doesn't quite fit.
So that led to a newer model.
Yes. The currently accepted model is the induced fit theory, proposed by Daniel Koshland in 1958. This is much more realistic.
Induced fit suggests flexibility.
Precisely. It proposes that the active site is not rigidly pre-shaped. It's somewhat flexible. It's the interaction with the substrate itself that induces a conformational change in the enzyme.
Ah, so the binding makes it fit.
Yes. The enzyme slightly changes its shape as the substrate binds, achieving the perfect snug sit. It's more like a glove molding around a hand than a rigid key in a lock.
And this induced change isn't just about fitting.
No, it's crucial for catalysis too. That conformational shift does two things. It optimizes the binding interactions, and perhaps more importantly, it brings those catalytic amino acid residues into the perfect positions to interact with the substrate, strain its bonds, stabilize the high-energy transition state, and ultimately speed up the reaction. The enzyme effectively hugs the substrate into reacting.
That makes much more sense dynamically. Okay, so within that induced fit active site, what are the actual chemical mechanisms speeding things up? The sources mention a few.
Right. That incredible rate enhancement, we're talking factors of 108 to 1012 times faster, isn't usually due to just one thing. It's often a combination of several catalytic strategies working together.
Like what?
Well, one common mechanism is acid-base catalysis. Amino acid side chains in the active site can act as proton donors, acids, or proton acceptors. Histidine is particularly good at this because its side chain pKi is near physiological pH, so it can easily switch between protonated and deprotonated states. This proton transfer can help stabilize charged intermediates in the reaction pathway.
Using protons to help the reaction along. What else?
Another major one is covalent catalysis. In this mechanism, the substrate, or part of it, temporarily forms a covalent bond with an amino acid residue in the active site.
A temporary bond.
Yes, forming a short-lived enzyme substrate intermediate. The classic examples are the serine proteases, like chymotrypsin or trypsin. A reactive serine hydroxyl group in the active site attacks the substrate's peptide bond, forming a temporary acyl enzyme intermediate. This intermediate is then broken down, releasing the product and regenerating the free enzyme.
So the enzyme gets chemically involved for a moment.
Exactly. And then there's something simpler, but still powerful. Proximity and orientation catalysis.
Just bringing things close together.
Basically, yes. The active site acts like a container. It binds the reacting molecules, substrates, close to each other and holds them in the optimal orientation for the reaction to occur. This dramatically increases the effective local concentration of the reactants and the probability of a productive collision. Just by doing that, you can increase the rate by hundreds or thousands of times.
So it's like forcing the reactants to sit next to each other facing the right way.
Precisely. The enzyme doesn't just lower the activation energy barrier, the tunnel through the mountain. It also makes sure the reactants, the hikers, are standing right next to each other at the tunnel entrance, perfectly aligned to go through. It's a combination of these strategies, acid-base, covalent proximity, that gives enzymes their phenomenal catalytic power.
That detailed mechanism flows really well into Section 4, enzyme kinetics and regulation. We've seen how they work. Now let's look at the math, how fast they work, and how that speed is controlled.
Right. Enzyme kinetics is all about studying the rate of enzyme-catalyzed reactions and how factors like substrate concentration affect that rate. When we plot the initial reaction velocity against the substrate concentration S, we typically get that characteristic curve.
The rectangular hyperbola. Starts steep, then levels off.
Exactly. At low substrate concentrations, the rate is roughly proportional to S more substrate, faster reaction. But as you keep increasing S, the enzyme starts to become saturated. More and more active sites are occupied. Eventually, adding more substrate doesn't increase the rate much because the enzyme is working as fast as it possibly can.
And that plateau, that maximum speed, is Vmax.
That's Vmax, the maximum velocity. It represents the rate when the enzyme is fully saturated with substrate. Every active site is busy processing substrate into product. An important point is that Vmax is directly proportional to the total amount of enzyme present. Double the enzyme, you double the Vmax.
Okay, Vmax is the speed limit. But what about that other key term, Michaelis-Menten constant?
This is arguably the more fundamental characteristic constant for a specific enzyme acting on a specific substrate. It's mathematically defined as the substrate concentration, S, at which the reaction velocity is exactly half of Vmax, and Vmax2.
Half the maximum speed. So conceptually, what does Ks actually tell us? What does it measure?
This is crucial. Km is an inverse measure of the affinity of the enzyme for its substrate. How tightly does the enzyme bind to its substrate?
Inverse measure. So low Km means high affinity.
Exactly. If an enzyme has a low Km, it means it only needs a small amount of substrate to reach half its maximum speed. It binds the substrate tightly, efficiently.
And high Km means low affinity.
Correct. A high Km means the enzyme needs a lot of substrate around to get to Vmax2. It doesn't bind the substrate very tightly, its affinity is weaker. So Km is like a fingerprint for that enzyme-substrate interaction under specific conditions, like temperature and pH. And importantly, unlike Vmax, Km is independent of the enzyme concentration itself.
Knowing Km helps predict how enzymes behave in the cell, where substrate levels can vary a lot.
Absolutely. For example, hexokinase in most tissues has a low Km for glucose, meaning it works efficiently even when blood glucose is low. Glucokinase in the liver, however, has a high Km, so it only really kicks into gear when glucose levels are high after a meal, helping the liver take up excess glucose. That difference in Km is physiologically vital.
Okay, that makes sense. Now, about that graph, the hyperbola is fine, but you often see the Lineweaver-Burk plot. Why the double reciprocal thing, 1V versus 1S, seems complicated.
It seems that way, but it's actually very practical. Trying to estimate Vmax accurately from a curve that just approaches the maximum asymptotically is tricky. You might never truly reach it experimentally.
Right, the curve just gets flatter and flatter.
Exactly. But if you take the reciprocal of both the velocity, 1v, and the substrate concentration, 1s, and plot them, the Michaelis-Menten equation transforms into the equation for a straight line, y equals mx plus c.
A straight line is much easier to work with.
Much easier. This linearization allows for a much more accurate determination of Vmax and Calmax from the graph's intercepts.
And what are the intercepts on a Lineweaver-Burk plot?
The y-intercept, where the line crosses the 1v axis, is equal to 1 Vmax, and the x-intercept, where it crosses the 1s axis, is equal to line 1 kilometer.
Okay, so you get precise values just by drawing the line and seeing where it hits the axis.
Precisely. And this becomes incredibly useful when we start looking at enzyme inhibitors, because different types of inhibitors change the slope and intercepts of the Lineweaver-Burk plot in characteristic ways. It makes identifying the type of inhibition much clearer.
Perfect segue. Let's pivot to regulation. The cell can't just let these powerful enzymes run wild. How does it control their activity, keep the cellular economy in balance?
Control is absolutely essential, and one of the most important mechanisms is allosteric regulation.
Allosteric. Allo meaning other, steric meaning site. So regulation happening somewhere other than the active site?
Exactly right. Allosteric enzymes are typically large, often multi-subunit proteins. They possess one or more regulatory sites, distinct from the active sites, called allosteric sites. Molecules called allosteric modulators, or effectors, bind non-covalently to these sites.
And that binding does what?
It causes a conformational change throughout the enzyme molecule, including altering the shape and catalytic efficiency of the active site. Positive modulators, activators, typically stabilize a high-activity conformation, often called the R or relaxed state. Negative modulators, inhibitors, stabilize a low-activity conformation, the T or tense state.
So binding here affects activity over there.
Yes. And kinetically, allosteric enzymes show a different velocity versus substrate curve, instead of the standard hyperbola.
They show that sigmoidal curve, that S shape.
They do, and that sigmoidal shape is highly significant. It reflects cooperative binding.
Cooperative binding, meaning?
Meaning the binding of the first substrate molecule to one active site on one subunit makes it easier, or in some cases harder, for negative cooperativity, for subsequent substrate molecules to bind to the other active sites on the other subunits. It's like the subunits talk to each other.
So the enzyme becomes more sensitive to small changes in substrate concentration around a certain threshold.
Exactly. It allows for a more switch-like response. A great clinical example is phosphofructokinase, PFK, a key control point in glycolysis.
Right, regulates glucose breakdown.
PFK is allosterically inhibited by high levels of ATP, which signals the cell has plenty of energy, and also by citrate, an intermediate from the TCA cycle. Conversely, it's activated by AMP, which signals low energy. This intricate allosteric control fine-tunes glucose metabolism based on the cell's real-time energy needs.
Makes perfect sense. What's another major control strategy?
Another critical one, especially for metabolic pathways, is feedback inhibition, sometimes called end product inhibition.
Feedback. So the end result affects the beginning.
Precisely. The basic idea is that the final end product of a multi-step metabolic pathway often acts as an allosteric inhibitor of the very first enzyme unique to that pathway, the committed step.
So if the cell makes enough of product E goes back and shuts down enzyme 1 at the start of the pathway, A, B, C, D, E.
Exactly that. It's incredibly efficient. Why keep the assembly line running if you've already got a stockpile of the final product? It prevents wasteful overproduction of intermediates and the final product, saving cellular energy and resources.
A classic example.
The synthesis of pyrimidine nucleotides. The final product, cytidine triphosphate, CTP, allosterically inhibits aspartate transcarbamylase, ATCase, the enzyme catalyzing the first committed step in that pathway. Simple, elegant control.
Definitely. And there's one more type of regulation mentioned, activating enzymes that are made in an inactive form.
Yes, control by activation of latent enzymes. Many enzymes, particularly digestive enzymes or those involved in blood clotting, potentially dangerous if active all the time, are synthesized and secreted as inactive precursors called zymogens or proenzymes.
What's pepsinogen?
Pepsinogen is the perfect example. It's the inactive zymogen secreted by stomach cells. In the acidic environment of the stomach, or by the action of already active pepsin, a small peptide fragment is cleaved off, irreversibly converting pepsinogen into the active protease, pepsin.
So they're kept safe until needed, then activated by cutting off a piece.
Exactly. It's often a very specific, irreversible proteolytic cleavage event that triggers activation only when and where the enzyme's activity is required. The blood clotting cascade is another complex series of zymogen activations.
Okay, that covers internal regulation beautifully. Let's move to section V, factors affecting activity and enzyme inhibition. We've talked about cellular controls, but what about external environmental factors, like temperature?
Temperature has a very pronounced effect. If you plot enzyme activity versus temperature, you typically get that characteristic bell-shaped curve.
Bell-shaped, meaning there's an optimum.
Yes. For most human enzymes, the optimum temperature is around 35 to 40 degrees Celsius, normal body temperature range. Below that optimum, the reaction rate decreases simply because molecules have less kinetic energy, fewer effective collisions.
And above the optimum, it drops off sharply.
Very sharply. As you increase the temperature above, say, 40 or 50 degrees Celsius, the enzyme starts to lose its specific three-dimensional structure. The heat energy disrupts the weak bonds, hydrogen bonds, ionic bonds, hydrophobic interactions that maintain the tertiary and quaternary structure. This unfolding process is called denaturation.
And denaturation means loss of function.
Complete loss of function. The active site geometry is destroyed. By the time you get to around 70 degrees Celsius, most enzymes are completely and irreversibly denatured and inactivated. This heat sensitivity or thermal ability is a key property.
And we use this clinically, don't we?
All the time. Think about it. Why do we refrigerate food? To slow down the activity of bacterial enzymes and prevent spoilage exploiting the low end of the temperature curve.
Right. And fever.
Fever is the body's attempt to raise the temperature to inhibit pathogen enzymes. But sustained high fever is dangerous because it can start to denature our own critical enzymes.
And in surgery, hypothermia.
Exactly. Induced hypothermia, lowering the patient's core body temperature during, say, cardiac or neurosurgery, reduces the overall metabolic rate and oxygen demand of tissues. This protects organs by slowing down enzymatic activity, giving the surgical team more time.
Fascinating how we manipulate that curve. What about the other major factor, pH?
pH also typically yields a bell-shaped curve when activity is plotted against it. There is an optimal pH at which the enzyme exhibits maximum activity.
And for most enzymes, that's around neutral.
For many intracellular enzymes, yes, the optimum is usually between pH 6 and 8, reflecting the typical pH of the cytoplasm. But there are very important exceptions you absolutely need to know for exams.
Like pepsin in the stomach.
Exactly. Pepsin works optimally at a highly acidic pH of 1 to 2, the environment of the stomach lumen. Conversely, alkaline phosphatase, found in bone and liver, has an optimum pH way up around 10 to 11.
Why such extreme differences? What's the mechanism?
It's all about the ionization state of the amino acid residues. especially those in the active site and those involved in substrate binding. Changes in H+ concentration, which is what pH measures, alter whether these residues are protonated or deprotonated. The enzyme's conformation and the charge state of the active site residues need to be just right for optimal substrate binding and catalysis. If the pH is too far from the optimum, the wrong ionization state can disrupt binding or interfere with the catalytic mechanism, like acid-base catalysis.
So the bell shape reflects the narrow pH window where the critical residues have the correct charge.
Precisely.
Okay, let's shift gears now to something hugely important in medicine: enzyme inhibition. This is the basis for so many drugs.
Absolutely. Enzyme inhibitors are molecules that bind to enzymes and decrease their catalytic activity. Understanding how they work is fundamental to pharmacology. We need to clearly distinguish the main types.
Let's start with one: competitive inhibition. The name suggests a fight.
It is. In competitive inhibition, the inhibitor molecule typically has a structure that strongly resembles the enzyme's normal substrate. It's a structural analog. Because it looks like the substrate, it competes directly with the substrate for binding to the active site. It physically blocks the substrate from binding.
So inhibitor or substrate binds, but not both at the same time?
Correct. Binding is mutually exclusive at the active site.
Right.
But the critical thing about classical competitive inhibition is that it's reversible, and importantly, it's surmountable.
Surmountable, meaning you can overcome it.
Yes. If you significantly increase the concentration of the actual substrate, the substrate molecules can effectively outcompete the inhibitor molecules for binding to the active site, and the reaction rate can eventually reach the normal Vmax.
Okay, how does this look kinetically? On that Lineweaver-Burk plot.
Kinetically, a competitive inhibitor increases the apparent coenzyme of the enzyme. Because the inhibitor is competing for the active site, you need more substrate to achieve half maximal velocity, Vmax2. So the coenzyme appears to increase, looks like lower affinity.
Carum increases. But what about Vmax?
Vmax remains unchanged. Because the inhibition is surmountable, if you pile in enough substrate, you can still eventually saturate the enzyme and reach the same maximum velocity as without the inhibitor. On the Lineweaver-Burk plot, the lines for inhibited and uninhibited reactions will intersect at the y-axis. 1 Vmax is the same, but the x-intercept, max 1 kilometer, will be different, closer to 0 for the inhibited reaction.
Increased carum, unchanged Vmax, and the clinical examples are huge here.
Massive. Many drugs are competitive inhibitors. We mentioned allopurinol for treating gout. It competitively inhibits xanthine oxidase, blocking uric acid production.
And the methanol poisoning treatment.
Right. For methanol or ethylene glycol poisoning, we administer high doses of ethanol. Ethanol competes with methanol, or ethylene glycol, for the active site of alcohol dehydrogenase, ADH. This slows down the conversion of methanol to toxic formaldehyde, or ethylene glycol to toxic oxalates, giving the body time to excrete the original poison harmlessly. Ethanol is acting as a competitive inhibitor antidote.
Brilliant. And statins.
Yes. Statins like lovastatin or atorvastatin are competitive inhibitors of HMG-CoA reductase, the rate-limiting enzyme in cholesterol synthesis. By blocking this enzyme, they lower cholesterol production in the liver. They are structural analogs of the HMG-CoA substrate.
Okay, that's competitive inhibition covered. What's 2? Non-competitive inhibition.
Non-competitive inhibition is fundamentally different. Here, the inhibitor binds to the enzyme at a site different from the active site. It doesn't typically resemble the substrate structurally.
Binds elsewhere, so it doesn't block substrate binding directly.
Generally, no. The substrate can still bind to the active site, and the inhibitor can still bind to its separate site, potentially forming an enzyme substrate inhibitor, ESI complex, or an enzyme inhibitor, EI complex. The binding of the inhibitor, however, alters the enzyme's conformation in such a way that its catalytic efficiency is reduced. It impairs the enzyme's function even when the substrate is bound.
So it affects the turnover rate, not the binding affinity.
Exactly. Because substrate binding isn't directly competed for, the chimerism typically remains unchanged in pure non-competitive inhibition. The enzyme's affinity for the substrate looks normal.
But Vmax.
Vmax decreases. Because the inhibitor essentially poisons some of the enzyme molecules, reducing the concentration of effective enzyme, the maximum rate achievable is lowered. Even at saturating substrate concentrations, the inhibited enzyme molecules just can't work as fast, or at all.
Unchanged carum, decreased Vmax, on Lineweaver-Burk.
The lines will intersect on the x-axis. -1 kilometer is the same. But the inhibited line will have a higher y-intercept. 1 Vmax is larger, meaning Vmax is smaller.
And examples? Are these usually drugs?
Less commonly designed as drugs, more often encountered as poisons. The classic examples are heavy metal ions, like mercury, Hg2+, lead, Pb2+, or silver, Ag+. These often bind tightly, sometimes quasi-irreversibly, to sulfhydryl Is groups of cysteine residues, which might be important for the enzyme's structure or function, but are often located away from the active site itself. This binding distorts the enzyme and inhibits activity non-competitively.
Okay, let's quickly touch on that related type you mentioned, sources sometimes distinguish. Uncompetitive inhibition. How's that different?
Uncompetitive is rarer, but distinct. Here, the inhibitor binds only to the enzyme substrate ES complex. It cannot bind to the free enzyme.
Only binds once the substrate is already there.
Yes. It essentially traps the substrate on the enzyme. Kinetically, this is unique because it decreases both Vmax and columnar, often proportionally. On a Lineweaver Burk plot, this results in parallel lines for the inhibited and uninhibited reactions.
Parallel lines. Decreases both Vmax and columnar. Noted. Now the really nasty one. 3. Irreversible inhibition.
Yes, irreversible inhibitors are often potent toxins or highly effective drugs. They typically form a strong covalent bond with a functional group in the enzyme, often within the active site itself.
Covalent bond, meaning permanent inactivation.
Pretty much permanent inactivation of that enzyme molecule. The inhibitor binds so tightly, often covalently, that it doesn't dissociate. The enzyme is effectively destroyed. The cell usually needs to synthesize new enzyme molecules to restore activity.
And the examples here are quite dramatic.
They are. Nerve gases, like disopropyl fluorophosphate, DFP, and many organophosphate insecticides work this way. They irreversibly bind to a critical serine residue in the active site of acetylcholine esterase, the enzyme that breaks down the neurotransmitter acetylcholine.
Blocking the breakdown leads to continuous nerve stimulation.
Paralysis. Yes, exactly. Paralysis and death. It's a potent mechanism.
But we use irreversible inhibition therapeutically, too.
We do. Penicillin and its relatives are classic examples. Penicillin acts as a suicide inhibitor. It mimics the normal substrate for bacterial transpeptidase enzymes, also called penicillin-binding proteins or PBPs, involved in building the bacterial cell wall. When the enzyme tries to act on penicillin, penicillin forms a stable, irreversible covalent bond with a serine residue in the enzyme's active site, permanently inactivating it. This blocks cell wall synthesis, and the bacteria eventually die.
So the enzyme essentially commits suicide trying to process the inhibitor. Clever. Any others?
Another well-known example is disulfiram, brand name Antabuse, used to treat chronic alcoholism. Disulfiram irreversibly inhibits aldehyde dehydrogenase, the enzyme responsible for breaking down acetaldehyde, a toxic intermediate in alcohol metabolism.
So if someone on disulfiram drinks alcohol,
acetaldehyde rapidly builds up to toxic levels, causing extremely unpleasant symptoms: flushing, nausea, vomiting, headache, palpitations. It's a strong deterrent based on irreversible enzyme inhibition. Aspirin also works by irreversible acetylation of cyclooxygenase, COX.
wow. Inhibition is clearly a powerful concept, both toxicologically and therapeutically. This brings us perfectly to Section 6, the diagnostic importance of enzymes and isoenzymes. This is where all the biochemistry hits the clinic floor. When you order blood tests, what are those enzyme measurements actually telling you?
It's a crucial application. We're typically measuring the activity, or sometimes concentration, of non-plasma specific enzymes in the serum or plasma.
Non-plasma specific,
Yes.
meaning they don't normally belong in the blood.
Exactly. These are enzymes that normally function inside cells in the cytoplasm or mitochondria of specific tissues like the heart, liver, skeletal muscle, pancreas, red blood cells, etc. They have very low concentrations in the blood of healthy individuals.
So finding high levels in the blood means
It usually signals cellular damage or death. When cells are damaged or lyses burst open, their intracellular contents, including these enzymes, leak out into the bloodstream. Increased cell turnover, like in some cancers, can also elevate levels. Measuring these enzymes gives us a window into the health of specific organs.
But how do you know which organ is damaged? Lots of cells have similar enzymes, right?
That's where isoenzymes, or isozymes, become incredibly important. Isoenzymes are different molecular forms of the same enzyme. They catalyze the exact same chemical reaction, but they differ slightly in their amino acid sequence.
Different forms,
Yeah.
meaning different genes or different subunit combinations.
Often different subunit combinations, or sometimes products of different but related genes. Because their structures differ slightly, they often have different physical properties like electrophoretic mobility, maybe different kinetic properties, CosMS, Vmax, and crucially, they often have different tissue distributions.
So measuring the specific isoenzyme pattern can help pinpoint the source of the damage.
Precisely. Let's look at the two classic examples taught in every med school: lactate dehydrogenase, LDH, and creatine kinase, CK.
Okay, LEH first.
LDH catalyzes the reversible conversion of lactate to pyruvate. It's a tetrameric enzyme, meaning it's made of 4 subunits. There are 2 different types of subunits, H for heart and M for muscle. These can combine in 5 different ways to form 5 distinct isoenzymes. LDH1, which is H44H subunits. LDH2, H3M. LDH3, H2M2. LDH4, HM3, LDH5, M44M subunits.
5 isoenzymes from 2 subunits, and their locations differ.
Significantly, LDH1, H4 is found predominantly in heart muscle and also in red blood cells in the kidney. LDH5, M4 is predominantly found in skeletal muscle and the liver. The other isoenzymes have intermediate distributions in various tissues.
Okay, so how does this help diagnose a heart attack, a myocardial infarction, MI?
In a healthy person, the serum activity of LDH2 is normally slightly higher than LDH1. However, after an MI, damaged heart muscle cells release large amounts of LDH1 into the blood. This causes the serum LDH1 level to rise significantly, becoming greater than the LDH2 level. This reversal of the normal pattern is called a flipped LDH ratio, LDH1, LDH2, and it's a strong indicator of myocardial injury.
Flipped ratio, heart damage. What about LDH5?
An elevated LDH5 level points towards damage in the liver, like hepatitis, or skeletal muscle, like muscular dystrophy or strenuous exercise.
Okay, LDH gives tissue specificity. What about the timing? And the other key marker, CK.
Right, creatine kinase, CK, also called creatine phosphokinase, CPK, is another crucial enzyme, especially for MI diagnosis. CK catalyzes the reversible phosphorylation of creatine by ATP, important for energy buffering in muscle.
And its structure.
CK is a dimeric enzyme made of 2 subunits. There are 2 types of subunits here, M muscle and B brain. These combine to form 3 main isoenzymes, CK1, BB, found mainly in the brain and lung. CK2, MB, found predominantly in heart muscle. This is the key one. CK3, MM, found predominantly in skeletal muscle.
So CK2, the MB form, is the heart-specific one.
Highly specific to the heart, yes. Normally, CK-MB makes up only a tiny fraction of the total CK in the serum. But after an acute MI, damaged cardiac myocytes release CK-MB. And here's the critical point for timing. CK-MB levels start to rise within 6 to 18 hours after the onset of chest pain. It peaks around 24 to 30 hours. This makes CK-MB the earliest and most specific enzyme marker routinely measured for diagnosing an acute MI.
Earliest marker, that's vital. So putting it all together, what's the typical enzyme pattern you see after an MI, the sequence of rises and falls?
Understanding this timeline is essential for interpreting results. Here's the classic pattern. 1, CK, total, and especially CK-MB, rises earliest, typically within 6 to 18 hours, peaks relatively quickly, around 24-30 hours, and importantly, it returns to normal fairly rapidly, usually within 2 to 3 days.
So CK-MB is the early warning signal, but it disappears quickly.
Exactly. If the patient presents late, the CK-MB might already be back normal.
What comes next?
2, AST, aspartate aminotransferase, which used to be called SGOT. This enzyme is present in heart muscle, but also liver and other tissues, so it's less specific than CK-MB. It starts to rise a bit later than CK-MB, maybe 8-12 hours after onset, peaks around 48 hours, and stays elevated a bit longer, maybe 4 to 5 days.
Rises after CK, stays up a bit longer, and LDH.
LDH total, and specifically LDH1, giving the flip ratio. Rises the latest, typically starting on day 2, 24-48 hours after onset. It peaks much later, often around day 3 or 4. But the key feature of LDH is that it remains elevated the longest, sometimes for 10 to 14 days or even longer.
So LDH is the late marker. If CK-MB is normal, but LDH is high with a flipped ratio, the MI likely happened several days ago.
Precisely. That pattern tells you about the timing of the event. Early on, look for CK-MB. Later on, LDH becomes more informative. Though nowadays, cardiac troponins have largely replaced enzymes as the primary MI markers due to even higher sensitivity and specificity. But understanding the enzyme pattern is still fundamental biochemistry.
Good point about troponins. Are there other key diagnostic enzymes we should mention?
Definitely. Marked increases in serum amylase and lipase are classic indicators of acute pancreatitis. These digestive enzymes leak out from the damaged pancreas.
Pancreas, amylase and lipase, liver.
For liver damage, ALT, alanine aminotransferase, previously SGPT, is considered more specific to the liver than AST. High ALT strongly suggests hepatocellular injury, like in viral hepatitis.
ELT for liver. And one more mentioned, acid phosphatase.
Yes, acid phosphatase, ACP. Specifically, the fraction of ACP that is inhibited by tartarate used to be a significant marker for prostate carcinoma, particularly metastatic disease, as the prostate gland produces high levels. Though PSA, prostate-specific antigen, is now the primary marker, ACP might still have some niche roles.
Okay, that clinical correlation is incredibly helpful. It really shows why understanding enzymes is so important for diagnostics. Let's wrap things towards the end now with Section 7, clinical significance and management strategies. We've seen diagnostics, but how does enzyme knowledge translate into treating patients?
Well, as we've already touched upon quite a bit, the principles of enzyme kinetics, specificity, and especially inhibition are the absolute bedrock of pharmacology. So many drugs work by targeting enzymes.
Right, we inhibit enzymes that are causing problems or maybe replace ones that are missing.
Exactly. We can inhibit enzymes involved in pathological processes. We already discussed using allopurinol, a competitive inhibitor of xanthine oxidase, to manage hyperuricemia and gout by reducing uric acid production.
We also mentioned statins inhibiting HMG-CoA reductase for cholesterol. What about adding enzymes therapeutically?
That's a major strategy, too. A classic example is in managing acute MI or stroke caused by blood clots. We can administer therapeutic enzymes like streptokinase or tissue plasminogen activator, TPA.
How do they work? They dissolve the clot.
They don't dissolve it directly. They act as enzymes that activate the body's own clot-dissolving system. They convert inactive plasminogen into active plasmin. Plasmin is a protease that then breaks down fibrin, the protein meshwork that forms the structure of the clot. So we use an enzyme to trigger the natural clot lysis process. It can be life-saving.
Using an enzyme to activate another enzyme cascade. Clever. What about cancer?
Enzyme therapy is used in some cancers too. For instance, the enzyme asparaginase is used to treat certain types of leukemia, like acute lymphoblastic leukemia, ALL.
How does that work?
Some leukemia cells cannot synthesize their own supply of the amino acid asparagine. They rely on getting it from the bloodstream. Asparaginase is an enzyme that breaks down asparagine in the plasma. By administering asparaginase, we deplete the external supply of asparagine, effectively starving those specific leukemia cells that depend on it, while having less effect on normal cells that can make their own.
Targeted metabolic starvation using an enzyme. That's quite specific.
It is. And reinforcing the inhibition theme again, think about anticoagulation therapy. Drugs like warfarin, related to dicumarol, act as competitive inhibitors of vitamin K epoxide reductase. This enzyme is needed to recycle vitamin K, which is essential for the synthesis of several clotting factors in the liver. By inhibiting this enzyme, warfarin reduces the production of functional clotting factors, thus thinning the blood.
So understanding the enzyme target allows precise therapeutic intervention.
That's the core principle. Whether it's inhibiting, activating, or even replacing enzymes, the detailed biochemical knowledge allows for rational drug design and therapy.
Fantastic. We've covered structure, classification, kinetics, regulation, inhibition, and the vital clinical links. You've really navigated us through the enzyme world. Time for a quick recap to help lock this in. Remember that mnemonic for the 6 major classes.
OTCLIL, oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases.
Perfect. Now, let's try 2 quick quiz-style questions just to reinforce some key concepts. Ready?
Go for it.
Quiz question 1, kinetics. We talked about killam reflecting affinity. So if an enzyme has a very low killam of value for its substrate, what does that tell you about their relationship?
A low killam means high affinity. The enzyme binds the substrate tightly and works efficiently even at low substrate concentrations.
Excellent. High affinity. Quiz question 2, clinical. Thinking back to the MI enzyme pattern, which isoenzyme is considered the earliest specific enzyme marker to rise in the serum after a heart attack.
The earliest one is CKMB, or CPK2. It starts rising within hours.
CKMB it is. Perfect.
Great job working through all that. You've just mastered another really key concept in biochemistry. Honestly, understanding the enzyme system, how they work, how they're controlled, how we measure them, it's like getting the keys to the whole cellular economy and how things go wrong in disease. Now, the next step for you is to take this detailed knowledge and constantly connect it back to the clinical signs, the lab results, the patient presentations you'll be learning about. Keep making those links.
Our deep dive into the world of enzymes is complete. We've journeyed from those fundamental protein structures and catalytic mechanisms right through to their use in diagnosing life-threatening conditions like heart attacks.
It's a huge topic,
Yeah.
but hopefully breaking it down made it more manageable.
Absolutely. And before we sign off, let's revisit that idea of enzyme flexibility from the induced fit theory and think about where it might lead. Here's a final provocative thought for you to consider. We spent a lot of time discussing how we use drugs to inhibit existing enzymes. But what about the flip side? What about designing better enzymes?
Enzyme engineering.
Exactly. If Koshland's model shows Enzymes are flexible and responsive, and we have tools like genetic engineering, specifically things like site-directed mutagenesis, where we can swap out specific amino acids. Imagine deliberately redesigning an enzyme's active site. Could we create therapeutic enzymes with, say, much higher specificity for a target, or maybe incredible stability so they last longer in the body, or perhaps even entirely novel catalytic functions?
It's not science fiction. We already have examples like engineered versions of TPA for clot busting that have longer half-lives. But think bigger. Could we design enzymes or catalytic antibodies, abzymes that specifically target and degrade the amyloid plaques in Alzheimer's disease, or enzymes that neutralize specific toxins more efficiently?
Or enzymes tailored to individual patient genetics for personalized medicine?
The potential is enormous.
Mm.
Using our understanding of structure, function, and kinetics to rationally design biological catalysts as therapeutic agents. That's really the cutting edge where biochemistry meets medicine.
Think about that potential designing molecular machines to fix biology as you continue your studies. It's an exciting time to be learning this stuff. We'll catch you on the next deep dive.